session this afternoon is bioinformatics analysis, pathways and beyond, and we'll also talk about some data integration. So finally, I mean, you've been doing all this sort of, hard work, collecting the samples, doing all this sort of, processing, talking to the core, getting, the data. So finally, you are at a point where you can start sort of, making some biological sense of the data and hopefully begin to answer the your biological or clinical question that you had in mind when you started working on it. And so in particular, in today's session, in this session we're going to talk about pathway mapping and enrichment. And so, as I said, so we're going to touch a little bit on the sort of most recent methods that go beyond biological pathways. And so, just quick word: the second session this afternoon, as I told you on the first day, you might remember, as really the goal is to give you a preview and set things up for the hands-on sessions that will take place tomorrow. So anyway, I think, to put it simply, many of you have experienced that either sort of, if you already had experience with metabolomics data or any other high throughput data, essentially, or, you might have been sort of, if you didn't, if it's not the analysis of your own data, maybe reviewing or reading papers, the manuscripts that other people published in the field. So it's simply no longer enough to provide the list of your differentially regulated metabolites. So basically, the quest is to be able to go from this to this, simply put. I mean, and in the process, obviously, to sort of, understand important biological implications, right? And so at a glance, it's easy, right? So, we know something about metabolism. Metabolism is understood, and you have seen in Gary and Patty's slides that he showed on the first day that, he showed this sort of, this sort of map, and then, the map of the unknowns around it. So so we already know that it's not so easy, and there are some sort of, implications and some caveats which George hinted at in his talk. So, when he talked about incomplete coverage of metabolism by these databases and so on and so forth. So at any rate, if we are sort of, I guess, if you're talking about pathway mapping or, we need a reference point, right? And so for those of you who are not familiar with this area, I mean, there are a lot, it can be very confusing because there are a lot of different databases out there. And so this slide is basically to sort of, simply help you get oriented within the sort of, variety of different databases that are out there. So for this particular session, we're particularly interested in various pathway databases, and I have a few more slides to talk more specifically to sort of, give some examples of the pathway databases, and you will hear a little bit more about various tools that make use of various pathway databases, and so on and so forth. The other type of databases that were touched upon and, talked about, there are various, and I mean, I guess, the reason I'm talking about these different type of databases because these are often sort of in different contexts, broadly referred to as metabolomics or metaboloid databases or something like this. And so I think, things that you need to keep in mind if you're looking for, like, the context in which I think Maureen mentioned earlier, that we're using a number of reference databases that contain sort of, various information about compounds, about spectra, about sort of compound structures, molecular weights, and things like that. So there's a number of those databases. There's a number of, like, compound databases that also have a lot of various information. In recent years, new type of, at least two new databases emerged which are basically the repositories for the experimental metabolomics data, and we are part of the NIH Common Fund Metabolomics Consortium, and so we're essentially obligated to submit the data sets that our core generates to the Metabolomics Workbench. This is the database that is being developed by the group at UC San Diego. And so it also was mentioned in one of Marine's presentations as well. Okay, and so these different databases are being used for compound identification. So, you know, a black box was mentioned in one of Charles's talks. So this is sort of, you know, this is my sort of, you know, black box, because I don't think, you know, there's something magical that has to happen here. Pathway mapping, you know, it's sort of gray, a little bit grayish. So we'll try to kind of, you know, talk a little bit more about this sort of, you know, pathway mapping idea. But first, just sort of a very quick look at the pathway databases. The oldest, I think, and perhaps the most, the best known is KEG database. KEG stands for Kyoto Encyclopedia of Genes and Genomes, and so it has the information about all these metabolic pathways. It's a great database. The sort of, you know, a little bit of a problem is the fact that several years back they lost their government funding, and so they had to go from a sort of, you know, completely sort of open and free model. Now, if you want to look things up on the web, you can still use it for free, but if you want to download the data and do something with their pathway data, you have to purchase a license. So that's sort of, you know, a little bit of an issue there. Just sort of, you know, a little bit of a closer look. Many of you can be familiar with it, even if you have not looked at the KEG website and haven't really explored the database itself. More often than not, you will see a picture of a pathway like this. And so I have a couple of examples, metabolic pathway here, and signaling pathway, MEP kinase signaling, I believe. So basically, you know, the sort of, you know, the simplest bioinformatics tasks that you might do, you have identified your differentially regulated metabolites. So what you do, you basically, you know, take this image and you just sort of, you know, put your differentially regulated compounds on this pathway, okay? So, you know, if you tried that, you know that it can be tedious and time consuming, and this is probably not the way to go, especially for a large metabolomics study, and especially for the sort of, you know, large data set. You probably want to do that sort of thing. Okay, so those of you who are sort of, you know, working in maybe like interested in microbiome and deal with sort of, you know, bacteria and so on and so forth, probably would be familiar with the biopsych databases. I mean, there's like a whole family of sort of, you know, databases and variety of microbial organisms and so on and so forth. And so these are being maintained by Peter Karp's groups. So the best annotated and the most well-known is EcoPsych for E. coli, and they have a number of different organisms that are annotated to sort of, you know, various degree there. So I think I just wanted to give you a flavor and kind of set the framework, mentioning a couple of these databases. And so another thing that I will mention, that in recent years, in the literature, a number of higher resolution, they're called metabolic reconstructions, and I'll say a few words about that in just a second, appeared in the literature, okay? And so these are two examples of human, high-resolution human metabolic networks, and they're called metabolic reconstructions, essentially. So the idea behind constructing these databases, just like KEG actually, is to use, in this case, human genome, for example, to help inform and sort of, you know, reconstruct and help reconstruct the metabolic pathways in humans. Okay, so we won't talk specifically, we won't talk sort of, you know, more about how this is done, but there is extensive literature and number of tools and for doing things like that. So it's important for me to mention this human metabolic reconstructions, because this is something that is really behind the tools that our group here have put together. And so tool that you're going to, those of you who attend the hands-on session tomorrow are going to get a chance to try it. If you haven't before, our tool is called Medscape, and so it is based on open source, widely sort of used software called Cytoscape for. So it's basically a tool for network visualization and analysis. And so what we did, we basically, took human metabolic pathways and build this in the Cytoscape world. This sort of, custom applications that are using Cytoscape, they're called apps. So Medscape is essentially an app for Cytoscape. And so what you can do with it, you can start with your metabolomics data or you can also have gene expression data. And Chuck mentioned it earlier in his talk. And so you can basically, map this data onto human metabolic pathways and reconstruct. And so the tool will construct the networks, and it will allow you to visualize changes in your data. And you have seen several examples of that. And I'll go over that in a little bit more detail in my presentation later this afternoon. So most recently, we added this correlation, so this module to Medscape for analyzing correlation networks. Again, more about this later, right? So, so what Medscape does is really, it takes your data and maps them onto metabolic pathways, right? And so what I often, and other people often heard or thought about, okay, well, great, you mapped my metabolites on, for example, okay, you mapped it to some kind of amino acid metabolism. So what? How significant is it? Is it important? Is it not important? That's a question that is very often asked. Okay, and so to answer this question, you basically, what you need to do, you need to put some kind of like, some measure of statistical significance on it, right? So it would be really nice to be able to rank these pathways. And this is sort of really the motivation behind, as George said, this is the motivation behind this sort of, pathway analysis tools. And this is the same review paper that George mentioned, which is sort of an excellent reference, which summarizes his methods very, very well. But in a nutshell, what you have to have your reference database. It can be a database of that maps your be they genes or metabolites or whatever you're trying to analyze, maps them to metabolic pathways or to biological pathways. These can be GOR terms or any other predefined biological categories as long as they're meaningful to researchers. Okay, and you have your input data, and then so essentially this review puts things into sort of, a little bit more of a historical perspective. But I mean, the first thing that people did, essentially, so if you have your list of differentially regulated metabolites, what you can do is you can basically compare that list to the sort of, your predefined pathways or other biological categories. And you can see if any of these are enriched or overrepresented. Any of the experimental genes are enriched within any of these biological categories. That's sort of, the basis for the this sort of, first level of analysis. Then the second level of analysis, which are sort of collectively called functional class scoring, is here. I mean, so maybe you don't want to look at your just sort of differentially regulated metabolites because it's challenging. Sometimes it's challenging to select the threshold. How do you know if something is just didn't make your cutoff? It still can be significant. In fact, you can have several metabolites that didn't quite reach statistical significance, but they're all changing in the same direction and they're all in the same pathway, right? And so it would be really nice to be able to somehow take that information into account, and that's what this sort of, additional type of methods are trying to do. And there is a number of tools that are doing that, in particular for gene expression data. And then, this is great, but none of these methods really take into account the pathway topology. And so this sort of, the latest sort of, version of these tools are trying to take that information into account. And so This is a little bit closer look at overrepresentation analysis. So basically, the methods that are often used for that, something like Fisher's exact test that was discussed in Shervin's lecture, I think, to some extent. So basically, you're trying to, so you have your genes or metabolites that are changing versus not changing. You set your threshold, and so you're trying to compare how many of these are in gene set versus non in gene set. So this is sort of this classical 2x2 table. And then, so you kind of use that information to estimate the statistical significance. And again, I'm sort of, repeating some of what George said at the end of his lecture just before lunch. The sort of, classical example of this sort of functional class scoring methods. So the idea there is to basically, start by computing gene level statistics. You can make, and there's a number of ways of doing that, as George said, but, simplistically, you can do a t-test and see sort of, have your p-values for your sort of, differentially regulated metabolites or genes. And then in step 2, you kind of aggregate this gene level statistics into sort of, pathway level statistics, and specific methods for doing that can differ depending on the sort of, ways of doing it. And you are going to hear some more about this. I'm just sort of so what I wanted to do is basically to set the stage for it. All right. And so next we're going to hear from Jeff about his tool, Metabolic Analyst. And many of you have probably, tried to use this tool. And some of these sort of, analysis, some of these enrichment analysis techniques are implemented in Metabolic Analyst, and I'll let Jeff talk about this. And the next talk this afternoon is going to be from Shizao Li, who is going to sort of, hopefully tell us a little bit more about his tool that was published several years ago, and I'm sure there are new developments. I cannot wait to hear about them. But so what this tool Novichok does is it tries to also sort of, incorporate the unknown compounds into analysis, and sort of, combine the pathway analysis with identification of unknown compounds. So we're going to hear more about this. And so finally, the third talk in the session, George is going to sort of, tell us about the new developments in the area of sort of, data integrations, and some of the issues that you need to be aware of when you're thinking about maybe integrating your experiments from different omics data. Okay, so I think I'll stop there.